Namaskar. Welcome to EPG Pathshala. In this module, we are going to address some of the issues related to spectroscopy, precisely atomic spectroscopy, X-ray and electrophoresis. First of all, we are going to have some introduction related to atomic spectroscopy. So, let us talk something about flame test, which is very prominent test which students undergo during their laboratories. When a sample of an clear alkaline earth metal is shown to the Bunsen flame, a characteristic color is seen. This experiment is precisely called as flame test. It has been used for many qualitative analysis for metal ions and comes under atomic emission spectroscopy, called as flame atomic emission spectroscopy. In the Bunsen flame, the metal ions are reduced into their corresponding atoms. The metal absorbs energy from the flame and rises to an excited state. This excited state in their atomic form. Then the atom comes back to its lower ground state in terms of energy by emitting the absorbed energy in the form of visible light of specific wavelength. There are some examples quoted on the screen which clearly show that when a metal ion, that is lithium, is shown to the Bunsen flame, it absorbs the energy and the color shown by the lithium is crimson. Similarly, strontium, when subjected to the flame, the heat absorbed and strontium also shows crimson color. Likewise, if potassium is shown to the flame, it gives violet color, whereas the sodium gives yellow color. Likewise, calcium and barium, when they are subject to the Bunsen flame, calcium yields orange color, whereas barium green color. They all fall in visible region, and it is corresponding to the kind of energy being absorbed. If it is very high, then definitely the light shown will fall in the lower energy band. This is very clear as we talk about the color due to the metal ions when subjected to energy due to heat. Let us have some introductory part about what is atomic spectroscopy. Atomic spectroscopy is the analytical technique used for the qualitative as well as quantitative analysis of the elements. This was first introduced in 1859 when Bunsen and Kirchhoff found that the radiation emitted from the Bunsen flame is due to the characteristic element present in the sample, as it is described so far. Atomic spectroscopy is precisely divided into two types. One is atomic absorption spectrophotometry, which is called as AAS, and the second type is atomic emission spectrophotometry, called as AES. Further, AES is divided into two categories: flame atomic emission spectrophotometry or flame photometry, and second type is inductively coupled plasma atomic emission spectrophotometry, which is symbolized as ICP-AES. Let us talk about atomic absorption spectrophotometer, that is AAS. AAS is an analytical technique that provides the quantitative information on elements present in a sample. It uses the characteristic wavelength of light absorbed by an element, which corresponds to the energy required to move electron from ground state to excited state, to measure the concentration of that particular element. Now, let us see how it works. Atoms present in different elements absorb specific wavelength of light, and this light of particular wavelength is analyzed to find the presence of that particular element. For instance, a lead lamp emits light from excited lead atoms, which is the standard wavelength to be absorbed by the lead atoms present in any sample. Let us talk about the principle on which AAS works. In AAS, first the sample is vaporized into the ground state free atoms called atomization, and then a beam of electromagnetic radiation emitted from excited lead atom is passed through the vaporized sample. Some of the radiation is absorbed by the lead atoms present in the sample. The absorption will be higher when there is a greater number of lead atoms present in the sample. The amount of light absorbed is directly proportional to the number of total lead atoms. A calibration curve is prepared by running several such samples with known lead concentrations following the same condition as in the case of unknown sample. The amount of standard absorbs is compared with the calibration curve to calculate the concentration of lead present in the unknown sample. Let us talk about some of the applications of AAS. AAS has been a very useful tool in many areas of chemistry, environmental science, pharmaceutical sciences, industry, etc. Environmental analysis, AAS help to monitor our environment, like finding out the levels of various elements in water, air, soil, that is surroundings of our nature. For example, the presence of elements like calcium, copper, iron, manganese, potassium, sodium, zinc and many other in soil can be analyzed using AAS tool. These metal ions are extracted using an extractable solution like 0.05 N HCl and 0.025 N H2SO4 or ammonium acetate from a known weight of air dried ground soil. The sample is then filtered and the extract can be directly used for the analysis of AAS to find out the level of the metal ions present in it. This method is useful to evaluate the nutrient quality of soil also, and further the data can be used in deciding the fertilizers needed for the soil cropping situation. That means we can enrich the soil which is running deficient of particular nutrient. AAS is useful to determine the elements present in natural water, sea and other saline water. It is also useful to determine the elements like cadmium, chromium, cobalt, copper, lead, manganese, nickel and zinc in airborne particles called metallic air pollutants. So, we can say that AAS is helpful to monitor the airborne pollutants and check their pollutants also. Let us see what flame photometry is. Flame photometry is one of the simplest atomic emission spectroscopic technique, which is very similar to the flame test as we have discussed so far while doing in laboratory the element detection test using Bunsen flame. It is useful for the detection of alkali and alkaline earth metals which has a relatively low excitation energy such as sodium, potassium, calcium, etc. How it functions is something very interesting to know. Flame photometry based on the fact that samples of alkali and alkaline earth metals undergo thermal dissociation when shown to a flame, and some of their atoms produced are excited to a higher energetic state. On return of these atoms to their ground state, they emit radiation which generally lies in the visible region of the electromagnetic spectrum. Let us talk about the principle on which the flame photometry works. Each element emits light of a particular wavelength. Within a particular concentration, the intensity of the emission is directly proportional to the number of atoms coming back to their ground state. In other words, this is proportional to the absolute quantity of the species vaporized in the flame, that is, the amount of light emitted is directly proportional to the concentration of the sample being used. The emitted light then isolated by an optical filter, and a photodetector measures the intensity of that light. The signal obtained upon further processing gives the detail of the required element. So, it helps in detection of the element qualitative and quantitative. Let us talk something about the application of flame photometry. Flame photometry has been useful as a primary analytical technique in many environmental studies, analysis of biological fluids, material science, etc. Recently, this technique has been useful and employed in determining the sodium content in seawater, used for analysis of Marina Beach water samples at Chennai. This technique has been useful in measuring the sodium and potassium level in the biological fluids such as blood, red blood cells, plasma and urine. Calcium content can also be determined in biological materials using this method. Determination of elements like sodium, potassium, calcium and magnesium in the plant tissues, for example sugarcane, can also be determined by using flame photometry technique. Inductive coupled plasma spectrophotometry, which is abbreviated as ICP. Let us have the introductory part. Argon gas, when flows through a copper foil connected to radio frequency generator, the slower central argon flow is covered by a stronger outer flow, and this slower moving argon gas become inductively coupled to the radio frequency region. The moving electrons and nuclei are ripped apart in opposite directions by the magnetic field, producing a gas called plasma containing both electron and positively charged argon ions. This is a very high temperature of about 5000 to 10,000 degree Kelvin. This plasma is useful for ionizing almost all elements of high excitation energies. How this ICP works? That means inductive coupled plasma spectrophotometry, how it works and what is the principle. When a high temperature ICP source combines with an atomic emission spectrophotometer AES, creates an ICP AES, and on combination with the mass spectrophotometer MS, create ICP MS. In ICP AES, the sample elements are quickly excited to their high energetic state upon giving plasma energy. When these excited atoms return to their ground's lower energetic state, they emit light of a specific wavelength corresponding to that element. The element is detected based on the characteristic wavelength of the light emitted, and the concentration of each element is analyzed using the intensity of that particular light. In ICP MS, the high temperature ICP source excites the atoms of the elements in the sample into their corresponding ions. Then these ions are separated and recorded by the mass spectrophotometer. Let us see the application of ICP coupled, that is inductive coupled plasma spectrophotometry. Recently, ICP spectrophotometry established as a cost-effective analytical tool technique used for multi-elemental analysis with a wide range of concentration from macrogram per level of major and trace element analysis in the field of environmental, agricultural and food, biological and clinical, geological and archaeological research. In case of environmental analysis, ICP spectrophotometry technique has a wide application in many areas such as soil, sediments, water, air, etc. When water analysis include determination of metals like arsenic, iron, chromium, cadmium, copper, manganese, lead and zinc in seawater, determination of phosphorus in municipal water, determination of heavy metals in city dust samples, etc. This technique is used to determine the metals and the metal compounds in suspended particulate matter in ambient air. For example, this technique has been used to check the suitability of the air in the workplace and occupational air exposure of workers. Let us talk about the next technique, that is X-ray, and how it is being used for the determination of various sample materials. In electromagnetic spectrum, X-ray region lies between 0.1 and 200 Armstrong. However, only a small portion of this total X-ray region, that is 0.1 to 25 Armstrong, is used by the conventional X-ray spectrophotometer, known as analytical X-ray region. Similar to that electromagnetic waves, X-ray travel in a straight line at a constant speed on vacuum, and they are not affected under the influence of the electric or magnetic field. However, they undergo reflection and refraction and can be superimposed to set up interference patterns. Interaction of X-ray radiations with matter can be explained as the interaction of X-rays with matter can proceed in three different ways. Number 1, absorbed or transmitted through the sample, that is medical X-ray used to see inside the materials. Number 2, diffracted or scattered from a crystalline material, that is X-ray powder diffraction, which we call as XRD, crystal X-ray diffraction, XRD, used to identify the crystalline material. And thirdly, causes the generation of X-ray of different color, X-ray fluorescence, XRF, used to determine elemental composition. So, XRF is the X-ray fluorescence, where the X-rays are used. So, let us talk an introductory remark on X-ray fluorescence, which is commonly depicted as XRF technique. An X-ray fluorescence spectrometer is mainly involved for regular and relative non-destructive determination of elemental composition of a material sample. It is used for analysis of rocks, minerals, sediments and fluids. There are two types of XRF spectrometers. One which is depicted as WD-XRF, that is wavelength dispersive X-ray fluorescence, and second is EDX-RF, which is energy dispersive X-ray fluorescence. So, XRF is based on principle. The fact is that atoms of a material sample, when excited by a primary X-ray radiation, as it is depicted in the picture on the screen, the electrons from the innermost K shells are released, and the resultant gaps are then filled by the outer shell electrons. During these transitions, fluorescent radiation is generated, which is characteristic of each element. This is read by the detector and gives the information on the composition of the sample material. So, from which outer shell, the electron is falling and excited rays are detected, that determines the characteristic sample. Let us talk something about application of XRF. XRF has a wide range of application in many different areas for both quantitative and qualitative analysis, such as environmental analysis, geology, recycling, etc. XRF photometer is used for quantitative analysis of determination of contaminants in solids, characterization of radioactive elements, for example uranium. It detects the composition of rocks and minerals, characterization of materials for recycling. It also suitable for the analysis of metals and metalloids present in dust samples. Given that children extensively use plastic toys, the toy industry is also very dependent on XRF technique so as to monitor the level of any harmful substances present as a content in the plastic toys. Electronic product manufacturers also get the help of XRF technique to monitor the compliance with the restriction of hazardous substance directive. It is used in determining the major, precious, trace elemental analysis and characterization of rocks, ores and soil. In watch and jewelry industries or in refining process of precious metals, XRF is needed to find out the accurate knowledge of the composition of the precious metal alloys to be refined. It is used for the identification of materials such as alloy and waste, further processing or recycling. What is X-ray diffraction, that is XRD? Let us have the introductory remark on XRD. XRD is again another analytical tool mainly used for the identification of crystalline materials and determination of unit cell dimensions. Max von Laue first explored this technique way back in 1912 and demonstrated that crystalline substances could act as diffraction grating for X-ray radiations. There are two types of XRD techniques: single crystal XRD and X-ray powder diffraction technique. We will talk about XRD. Single crystal XRD is used for the determination of unit cell dimension and the position of the atom within the crystal lattice. This is particularly useful for characterizing of new materials and to find out the answer regarding the molecular rearrangement of the crystalline substance. X-ray powder diffraction is recognized as one of the prominent technique in the field of environmental sciences, geology, material science and engineering for rapid identification of crystalline substances. In this case, the sample need to be pure, finely ground and homogenized for finding out the bulk composition. It is further useful for the detailed characterization of crystalline substances and detection of unit cell dimension. Identification of fine grained materials is also carried out using this tool. Let us see some of the major applications of XRD. XRD has many applications in the field of environmental science, agriculture, geology, material science, chemistry, forensic science and the pharmaceutical industry and many such more industries. In the environmental studies, XRD technique have been used to quantitatively analyze the amount of amorphous substances present in the particulate matter. XRD is useful to study the areas affected by acid mines, drainage to detect these secondary minerals and fine grain precipitates. We will see about some of the major significant points regarding electrophoresis. Frederick Rios in 1807 first demonstrated that dispersed clay particles in water migrates under the influence of an electric field. This is due to the presence of charge interface between the clay particles and water, and the rate of migration particularly depends on the strength of the field which is applied, the net charge, size and shape of the molecules, and the ionic strength, viscosity and temperature of the medium in which the particles are moving. The above process is termed as electrophoresis, the movement of ions in a solution under the influence of a uniform electric field. Electrophoresis is used to study the properties of a single charged species and as a separation technique for separation of molecules by size, charge and binding affinity. Arne Tissels bagged the Nobel Prize in Chemistry in way back in 1948 for his work on electrophoresis and absorption analysis, particularly for his discoveries on the complex nature of serum proteins. He examined horse serum and observed it as a mixture of 4 components, 1 albumin and 3 globulin. As it is clear in the picture given on the screen, the migration of ions in electrophoresis chamber, how it occurs. You have a background electrolytic cell and the sample is dispersed in that. If we put this sample in the applied electric field, then the migration of the ions on the either side of the material occurs, as it is clearly denoted in the figure. You can see that the negative charge on one side and the positive charge moves on the other side. So, what is the principle on which electrophoresis works? When a mixture consisting of electrically charged molecules kept under an influence of electric field of strength E, the molecules will flow towards the electrode of opposite charge, as it was shown earlier in the figure. However, the velocity of each of the molecule will be different because of the negative and positive charges and depends on its own physical characteristics and the experimental conditions. The velocity of movement, that is V, of the charged molecules in an electric field can be very well expressed by the equation nu is equal to E into charge, that is Q, upon F, where F is nothing but frictional coefficient defined as frictional resistance to the mobility of the molecule, where Q is the net charge determined by the number of positive and negative charges present in the molecule. Let us talk about the different terms which are being used in the electrophoresis, such as paper electrophoresis. It is generally used for the analysis and separation of the small molecules such as amino acids and nucleotides, not suitable for proteins. In this case, the paper is moistened with buffer and placed between electrodes in electrophoresis chamber. The second form of electrophoresis is capillary electrophoresis. In this case, the material and the electrophoresis medium are placed in a long, fine bore capillary tube. A very small amount of sample in nanoliter is kept at one end of the capillary and subjected to electrophoresis. This method is suitable for separation of very tiny molecules or almost negligibly seen by naked eye, that is DNA molecules, of size as small as single nucleotide. Another form is gel electrophoresis. In this method, separation occurs in aqueous buffer supported within a polymeric gel matrix. This technique has many advantages than the above, such as larger samples can be accumulated for preparative scale electrophoresis. The nature of the gel matrix can be varied to fit for a particular use. There are many applications of electrophoresis technique. In case of environmental monitoring, electrophoresis is an effective tool mainly used in clinical laboratories to separate proteins. In addition, recently, particularly capillary electrophoresis, it has shown to be useful in organic pollutant analysis such as pesticides, phenols, pharmaceuticals, nitroaromatic compounds, amines and many such molecules. This technique is also useful for trace metal analysis in many environmental analysis. Source tracking contamination in food and other materials can be helpful by electrophoresis technique. When a particular microbial organism contaminant is found in a certain product, electrophoresis can be helpful to track this organism to its source. This is specifically important in food, pharmaceutical and various manufacturing system industries. So, with this module, what we have seen that there are many techniques which are helpful for monitoring environment as well as many other industries which are useful in day-to-day life, such as pharmaceutical, then some industries related to food or maybe medical, and the tool we talked about were atomic spectroscopy, XRDs, then flame photometry, electrophoresis. Thank you so much.